Right. Sorry, I'm still slightly on the airplane, but I'm definitely here. So thank you very much for the invitation to come here to present some of the work we've been doing at Tate over 2 decades these days. And sorry for being very, very late. My flight got cancelled yesterday, so I just pretty much arrived at 2 o'clock. So managed to hear some of the talks, which have been really interesting. Particularly for me, hearing that Shell Sol is green, I'm like, okay, I need to think about that a bit more. Hey, Piero. I'm going to be talking about various things, including the GreenArt project, of which Tate is a member. And I'm also the sort of task lead for the 2.3, I think, task, which is just gathering all the heritage partners together around the cleaning work package, which has been excellent fun. So just to start a little bit on this, Tate in London has 4 sites, just in case you don't know it, and 2 stores, and is one of the massive collections, and our modern and contemporary art collection is growing exponentially. So when I joined Tate, I was charged with taking on work that Tom Lerner had started while he was at Tate, and it's pretty much mushroomed into many different projects that I've had the pleasure of doing and being involved in and working with a lot of brilliant people. So what I thought might be helpful, I hope it's helpful today, is to talk about some of the projects that had case study conservation treatments embedded into the research model, which I think might be helpful. I don't know, I certainly hope so. Of the projects, I really haven't got enough time to go into much detail, of course, but of the projects that are outlined in orange font, they're the ones that have had case study works of art conservation treatments. Some of them were planned, some of them were definitely not planned. And on the right-hand side is just acknowledging all the PhD students that have been involved in some of these projects and are still carrying on with work around cleaning science. So some of the challenges of modern painted surfaces, and I'm sure you've probably been talking about them all this morning and I missed it all, but I just thought I'd throw this painting up as an example of the kinds of things we are encountering in the museum these days, and this is an iconic painting in Tate's collection, Roy Lichtenstein's Wham, which we cleaned in about 2017 as part of the NanoRestart project. And why this painting was problematic was for a number of reasons. First of all, there are several paint types. There's oil paint, there's mixed oil and magna, there's an alkyd priming, and there's magna paint. There's no acrylic emulsion paint, which was helpful, but in the end, it was quite tricky for this reason. The speech bubble at the top, now marked in blue, is highly water sensitive, so one swab roll and the paint was gone. The background, which is a Ben-Day dot stencil, the stencils in the background are oil paint, which was mildly to medium water sensitive. All the coloured parts that are marked in purple are actually acrylic solution magna paints, which are mineral spirit soluble, and then the entire painting was sensitive to mechanical action. Swab rolling. Is this working? Ah, brilliant, thank you. I can move now. Right, so you can see why we might have had to reach for different cleaning methods. So I kind of, when I was thinking about what to present today, I kind of thought it might be helpful to see how we might approach the methodology of bringing all this research together and also then translating it to cleaning a work of art, which is not an easy task. And we're still learning how best to do that, of course, especially when novel materials or novel techniques are in the mix. And of course, I always say that all of these different types of research feed into each other. So you have the research on paint materials or whichever material, whether it's plastics, whatever it is you're looking at, which involves all sorts of different parameters and art history, technical art history, talking to artists, all of that kind of thing. Paint properties, which I consider kind of bulk film properties, and surface properties. Then you have the response of the material to the treatment, which could be to solvents or it could be to atomic oxygen. And then you have the cleaning system development and modification and redevelopment and moving on. And then the assessment and the comparative studies. All of that, to me, needs to happen and needs to come together in some helpful way. And then on top of that, you've got kind of have the wider things that are influencing the decisions we make about research, which are, of course, the funding sources. And again, thank you for the green chemistry call. Institutional values, again, if you're working in a museum, anywhere really, you have to do a lot of internal influencing as well about what treatments to undertake and to justify the resources involved. The economic crisis and, of course, why we're all here, the climate emergency. So all of those things come into the mix. Complicated, but a very rich place to be exploring. And then there are different stages to the case study-led research. I thought I'd just throw these down here. I'm not going to go through them all, but obviously defining the research that you're going to do within as wide a context as possible is really important. Characterising case studies using conservation, technical and analytical techniques. Preparing mock-ups if you're choosing to go down that path. And then having a dialogue with the work of art, I thought I'd say, because often you're looking at, if you're using a case study work of art, you have to go backwards and forwards to the artwork and then adjust your mock-up testing accordingly and vice versa. It's a two-way street, it's a conversation. You discard systems along the way and you have to justify why, and you spend the rest of your time evaluating systems on mock-ups and then taking them to the artwork. Eventually, if all is well, all are in agreement, you get to the case study conservation treatment, during which time further adjustments are always made because conservators are highly skilled at using different materials and techniques. And then the scientists are around pre, during, and post for looking at assessment of change. And then the whole conversation goes, well, what does that change mean? So you can see, well, I've got lots of grey hair. So within that context, I thought it best to talk to you about GreenArt at Tate. We are one of the members of the GreenArt project, as I've mentioned. And this is the team who may or may not be listening at the moment over in the UK. There's myself, Katie next to me. There's a Bridget Riley painting in the middle. There is Anna Cooper at the other side, and there's Marana Novak, who's the conservation scientist employed for GreenArt at Tate. And up in the top corner, I've hovered Annette King into the picture because Annette has now taken Anna's place because Anna has had a baby and is having a well-earned rest. Bottom right, we have Piero himself and David, who were at Tate and look very, very happy to be looking at the Bridget Riley painting that we've chosen to work on. So, Bridget Riley, why did we choose Bridget Riley for our case study conservation treatments? First of all, she's a very well-known painter. She's still around. She's very, very engaged in the technical side of things. And both these works have been flagged for cleaning for some years. They're both polyvinyl acetate and they're both on hardboard. So Tate has spent quite a lot of time researching acrylic paints and helping with oil paints, but we haven't really focused on polyvinyl acetate. So it was an area of a gap in our research. As you can see, lots of analysis, not very interesting materials, don't tell Bridget that, but they're all fairly straightforward, except for the polyvinyl acetate. I don't need to tell anyone in this room all the types of research you have to do when you're anchoring a case study treatment into your research. But of course, there's a technical art history side of things. There's conservation records, and at Tate, they're often 50 or 60 years old. These paintings, or at least one of those two paintings, was in fact acquired quite quickly after it was painted. So we've had it for as long as we've had it for the same period of its age. Conservation records, curatorial records, looking at our interviews with the artist, and also, luckily for us, we were able to ask Bridget Riley in to Tate. And here she is, looking at Hesitate on the left. And there's me on the right, feeling rather nervous that I'm really close to Bridget Riley. And in the middle, we have two of the Tate team. That's Jane McCree, head of paintings, and Anna with Bridget. And there is Phil Young, who is a modern contemporary painting conservator in London of great experience, but is also chair of the Bridget Riley Art Foundation. So we're very engaged in the Bridget Riley Art Foundation, and obviously she knows what we're up to, and she's very happy for us to be doing what we're doing. They're not going to endorse anything that we do to her paintings, so they're having a step back from that, but they're very engaged in what we're up to. So, as always, people analyse artworks. We go through this process ourselves, sometimes many times, depending on how many projects we are looking at and how many requests we have from the conservation teams. But we've recently had quite a bit of input in terms of equipment, portable equipment, which has been really helpful because we were struggling with that in the past through a couple of grants. And here are just some images of various scientists and conservators using various techniques, whether it's high rocks, Joyce Townsend doing a good old-fashioned cross-section there, XRF, portable FTIR, everything we could throw at it, we throw at it. We also take samples and do pyrolysis GCMS and other types of more sophisticated analysis. So in the background, Marana has been doing a literature review on what we know so far from the field about polyvinyl acetate paints, how they respond to aging, how they respond to solvents, etc. So we published this paper last year as a kind of, I guess, forming a groundwork for some of the analytical work that's running in parallel to the conservation work. And that was quite interesting in terms of identifying gaps in research. Following from that, Marana, apart from analysing the paintings, Marana has been, I guess, optimising a pyrolysis GCMS method for the analysis of polyvinyl acetate paints and the copolymers as well. We're having a particular emphasis on the additives because some of the additives move to the surface and some of the additives are extractable with solvents and maybe other techniques. So that's going on in the background. So then I get to the question of mock-ups. Again, there are a lot of decisions to make about mock-ups, and when you're making them for a case study, they get more complicated. You have to do all of that analytical work, all the cross-sections, all the looking, all the imaging, in order to make them as close as possible, knowing full well that it's a compromise. It also depends on what analytical techniques or imaging techniques you're going to be using. Sometimes you have to tailor a mock-up to a particular analytical technique or an imaging technique, for example. But what are they good for? You've seen a lot of mock-ups already today. I saw one in the talk that I've seen so far today. They're really, really helpful on so many levels, but they're not perfect and they're not representative of an artwork, unless it is an artwork. But what they are really good for with comparative cleaning studies is two things. One is trying out the systems and doing a lot of mistakes, pushing the systems to their limits, finding out how to use them properly, and to develop the skill sets required to manipulate the materials, the systems, the machinery, whichever cleaning system you're using. That's incredibly helpful, and no matter what the results are, conservators who are using the mock-ups end up feeling more confident when they go to the artwork, amongst other things. So the mock-ups for the Riley painting, we're doing kind of a combination of Hesitate and Fall, which are the two works we're looking at. As I said, they're on hardboard. So we do a whole bunch of things, kind of rough mock-ups and some that are a bit more sophisticated. So the rough ones are on the left there. The paintings are either black or white or shades of grey. And on the right-hand side are the drawings for the more sophisticated mock-ups that are tailored to the paintings themselves. And here they are in their semi-finished state. We have here Anna and Katie, the conservators, were in charge of making the mock-ups with a lot of support from the science team. And Anna was the one who did most of the painting. And we based the mock-ups around either paint features of the artwork or condition features that the artworks are displaying. So scuffs and marks and lips and all sorts of different things which might prove problematic for cleaning. And then, of course, the famous soiling, which Tate did not invent, I just like to say. It was a Richard Wolbers publication. But we have tweaked it several times and used it in several projects. And here we have Helen Brett spraying, just using an air gun, some of the soiling that we use onto the mock-ups, which was quite an eventful day. They were aged in bezo conditions, light aging without UV in one of our chambers. And then you can see the soiling levels. We tried to match the soiling levels with similar to the painting soiling level. So having done all of that work, which is actually quite a lot of work, as you can imagine, we then think about cleaning. And because we're in GreenArt, of course, we're going to be looking at the new GreenArt materials as they come through. We're primarily looking at soiling removal, as you would have noted. And so we are looking at aqueous systems in various shapes and sizes, primarily. And so what I've generally done is taken the approach that we've been using for some time, which was developed over many years of research, but you could consider it the cleaning acrylic painted surfaces approach or the modular cleaning program approach, so quite well-known approaches in conservation these days. And choosing systems. We were starting with water and we're building up cleaning power, we're adjusting pH, we're adjusting conductivity, we're moving up in strength, we're backing off when we need to, we're changing pH. If it's too strong, we're getting too much pigment pickup, we're doing all of that stuff on the mock-ups, writing it down, taking photographs, and making beautiful PowerPoint slides, hopefully. And then once we get through that sort of initial phase of kind of just throwing things at the mock-ups, really, we end up in a place where we're being a bit more careful and a bit more sophisticated, and we start to use star diagrams, which I first came across with the dry cleaning research at RCE. And they've proven quite interesting in the last 10 years or so in our work, because we are able to sort of tailor the star diagrams according to the treatment, according to what you're looking at. So there are many conversations that happen about what parameters are important to look at during the cleaning. And it changes with each artwork. There are some that are quite always the same, like cleaning efficacy or pigment pickup or gloss change or whatever. But there are some that are really relevant either to the cleaning system or to the artwork that you need to bring in. And the most challenging part, and I don't know if anyone else has had a go at this in this room, but is defining what the scales are, and also describing what the scale points are so that it makes sense to someone else. Quite hard to do. So we're at that point now with the GreenArt project at Tate. We've got our mock-ups. We are going through the process of tailoring water, moving to spreadable gels, aqueous-assist hydrogels, and then we're going to start working with the new GreenArt materials which have come through, which I'm sure Piero has already described to you. So we're at that stage where we're really starting to get quite refined on the mock-ups, moving closer to the artwork. But another big part of the work that I've been trying to do, at least at Tate for a long time, is to try and characterize the effects of cleaning, and even harder, put that into context. So measuring change on any level, whether it's removing materials from paints with solvents or with other things, or it's changes in gloss. All those things are quite difficult to actually capture, but even more so to kind of put into context or give meaning to. I've been working with many people, trying to translate that kind of science-measured change into something that's meaningful for risk, for informing risk around conservation treatment. All those techniques and things like that we've done several times on artworks and on mock-ups, and again, there have been others mentioned already today since I've been here that are also really, really helpful, and it'd be really interesting to have a session on evaluation of cleaning effects, I would think. Some of the artworks we've looked at, Tate's collection is very rich and also full of artworks that are not protected, not varnished, and nearly always on the wall, so they're getting quite dirty at this point in their lives. I'll come back to a couple of those in a second. One of the other curly questions is residues, and that may or may not be relevant to Moxy, for example. There might be different kinds of changes that you're looking at, but for sort of wet cleaning systems and anything that involves solvents or other materials, residue studies are important for looking at risk and looking at legacy of treatments. And again, we've used many things over the years for that. And I very much realise that no matter how, if you look hard enough with the right technique, you're going to find something. Ilaria is nodding, yes. So what does that mean? What does a monolayer of surfactant mean on an artwork that's going to be RH, temperature cycling, pollutants, particulates, people, rain, whatever? How does that compare? And that's one of my reflection points, possibly for discussion later. It's very hard to inform that, and I've had many a conversation where I've ended up slightly confused, even after doing all the science. XPS is another technique, just sort of throw that up, was part of the CMOC project, where we looked at silicon residues from the siloxanes, and we looked at sodium residues from sodium dioctosulfosuccinate surfactant, both of which we found some. Yeah, we found some. So, reflections. Where am I at with my timing? Am I all right?
You're good, yeah.
I'm good, thank you. I've got about 6 reflections, I think, which are kind of hopefully conversation starters, hopefully not conversation killers. We'll find out. I just thought, well, what's been really important in all of these projects for me and for the teams I've worked with over the years? What have been some of the key things that have been important for that research into practice translation step that we all need to do for things to become used, important and significant in the field? Collaboration and teamwork, hugely number one. You can't do it on your own, and if you do, it's going to have gaps in it. Conversations are much better with other people, and I mean all of the people, as in inventors through to conservators and technicians through to scientists and everybody else in the mix. Always hugely important. And for me, within the Tate context, it's about funding conservators to do the research, taking them away from their everyday jobs and letting them focus. And I think that needs to happen more in projects. That's certainly how we worked in NanoRestart and how we've worked in GreenArt, and it's been hugely productive in lots of ways. The other things, and I know you guys do workshops, but the other thing that's been really helpful for this kind of research arc, if you like, is doing workshops for conservators, for conservation students, because they ask really great questions. They push your materials and go, that's rubbish. I'm going to keep using saliva, thanks. And you go, okay, tell me more about that. So you can have those conversations, and they're really important to help you as a researcher to go, oh, mate, I'm not in the right place here. I'm being too sciencey. I'm not answering their needs. I'm not addressing their needs. So that has been hugely important, and I've done them since about 2005, and I'm still doing them. Every year, maybe 2 or 3 times a year. They're really, really helpful for getting feedback as well as getting your systems out. Sorry, microphone. The other thing that I found helpful, bearing with conservators as the primary audience for my research, my as in everybody that I'm involved with research, is trying to do science that might look a bit basic, but actually directly speaks to conservation practice. Okay, so things like swelling studies. What do these paints do? And this is an example here of looking at, well, there's been no swelling studies on polyvinyl acetate paints where we're looking at pH and conductivity effects. Can we extrapolate the acrylic paint research, or is it going to be different? Simple question. People don't know what to clear their systems with, etc. So here we have, I just pulled out some of the data the other day, the difference in pH causing differences in swelling. So we will publish this once it's finished, but trying to remember that conservators are dealing with practical things and they need practical help. I've never been able to produce a decision-making model for this kind of work. It's too complicated in my head anyway. But what we have been doing is retrospective maps of the decisions that we made. So at the end of the treatment, we try and map how we got to where we got to, so that conservators may find that helpful, especially if they're picking up, say, PEGI gels for the first time, or the GreenArt gels for the first time, or something else, atomic oxygen for the first time. It's helpful to do this kind of thing, and this was the one we produced for WAM. And this is a more complicated one that PhD student Liana Pozmo-Stovland did, which might look a little bit more like the type of map that atomic oxygen users might produce, because this one's got different types of non-contact modes as well in it. She was looking at the Munch-Aula paintings in Oslo, and her PhD was really about this process, making the mock-ups, testing all these different systems, and then coming up with a map of how she got to where she got to. They're both published in Heritage Science if you're interested in them. One of the joys of being ancient in a museum is occasionally you can come back to treatments that you did or worked with others to do 10 years ago or so. And this was published in ICOMOS in Beijing, 2020, 2021, due to good old COVID. So what we did here, the main research question was, When we cleaned these paintings with water and a swab back in 2007, we removed all of the migrated surfactant because we did a hell of a lot of science to show it was there. We then removed it, we showed it had been removed. The question was, has it come back again? Can we see it? Has it re-emerged? And has it re-emerged within the context of the storage conditions that Tate has? Because there's been a few publications about storage conditions for acrylic paints and surfactant migration, and I was curious about, are we seeing what we expect to see or not? And the answer is, there's no surfactant on the paintings we looked at yet, which does slightly contradict the environmental research that's gone on, which just means we've got more work to do. And finally, who'd have thought soiling can be sexy? I never thought it could be sexy. It just takes one young, enthusiastic social media person to turn something that can be rather dull into something that gets a lot of hits on Instagram and whatever. And this was just really a note to self to not be so boring about things, that people who aren't terribly involved in this stuff can find the tiniest bits of it fascinating. So don't forget to talk to other people who know nothing about what you're doing, and then they might just come up and find soiling really sexy. So there you go. Thank you very much. Probably me.
Okay, thank you so much for that interesting talk that touched on so many important processes that we're all going through in all of our projects. I'm sure there are questions in the room. Does anybody have a question for Bronwyn?
Oh, Cecil. Hello. Oh, it's going to be a hard one, I can tell by your face. Oh, no.
Ready? Okay.
I'm ready.
So, the star diagram.
I've
tried to actually use it as decision-making help for conservators in general, and you know, reinventing it every time for different purposes. And I find it really useful, so thank you for promoting it. And I was just wondering if you have experience with that, now that you talk about how do we bridge the gap between research and practice. Do you sometimes use it, or do you promote it with conservators to use it as an everyday decision-making tool?
I think the short answer is, and I'm feeding back, is, move that away, where it's relevant and where they've got the time to think about it, yes. It's not always relevant, and they don't always have the time. So definitely when you've got funded projects or PhDs where people can really mull over this, definitely. And I think probably we will start using it more. I wanted to bring it into workshops, but it needs a while to actually create them. So you need a bit more of a workshop time to do that. But it's in essence, yes, but not on an everyday level yet. Yeah. It's good to hear you've done that, though.
Any other questions for the moment? I'm sure you'll be overwhelmed with people coming and asking you directly.
Pleasure.
One more?
One more?
Yeah. Hi. I have listened to you often say you are a scientist, a conservation scientist. You are also, or at least you have been a conservator.
Yes.
And it's very interesting in your presentation, the approach towards the artist. It has been always bigger and deeper.
Oh, Elena, what is your exact question? Has it always been that involved?
As you have evolved this approach towards the artist, the way you have done science, conservation science, and the way you see the work of your colleagues has evolved, has changed. In which sense?
Yeah.
If you can do your dots.
Yeah, I mean, phew, having been a scientist and a conservator, but I'm a conservator who worked for several years as well, so I've had the joy of being confused at the surface and having victories, being plagued with questions. I guess I approach the science that way. I might not have fully got your question, so come back at me if I haven't, but for me it's important to have all of these aspects in the mix, especially when you're getting to the research into practice side. Slightly controversial, some scientists don't do that terribly well. Not in this room, of course, but it's been really important because I think based in a lab within a conservation department, you have to engage, you have to be relevant, you have to influence, and you have to deliver. So I think all of that's been in the mix as well for me. But I am genuinely interested in that interface, that cleaning interface, the treatment interface. I've always been interested in that. And the surface, and what the surface looks like, what it looks like now, what it might have looked like, what change can happen with treatment apart from getting the soil off, what else kicks in. All of those things are really interesting questions and very hard scientifically to answer, I find, if you've got really simple ways of doing it. I'm really happy to hear that. I don't know if that answers any of your question well enough, Elena. The role of the artist, perhaps?
Well, a sentence, your sentence, when you were approaching Rothko,
Oh, yeah. Yep.
you said sometimes all this knowledge, scientific knowledge, can be a problem to arrive to the artist and the essence of the work of art.
Oh, yeah, I remember. Oh, yeah. Did you listen to the press conference? I remember saying that having known huge amounts about the constituents and the way the painting was made, it's still a mystery to me. And I love that. And I don't know what that makes me, a scientist or not. I don't know, but I love the fact that there are still things in that creative process that we can't unpick. Is that what you're getting at? Yeah, okay, yeah. I like that space.
With that, I guess we'll close to talk.